Today we are going to start the GI system revision series. So let us start with GI secretions, the main secretions from our GI system. So first we have the introduction to GI secretions. So mainly why GI system is for digestion and absorption of food. So the GI secretes like about, you can imagine 9 liters of secretions per day, and out of which of course again 8.8 liters per day is again reabsorbed back. So only 200 ml is excreted in the stool. So remember, 9 liters per day GI secretions, in that 8.8 is reabsorbed and only 200 ml is getting out from the stool. So the secretions, what are the GI secretions starting from the top to the bottom? From the top, of course, in your mouth, the saliva, and next gastric secretions from our stomach, and next the exocrine pancreatic secretions of course from the pancreas, next the biliary secretions from the liver, and the small intestine secretions from small intestine. Large intestine don't have that kind of secretions. Large intestine only secrete mainly mucus. So the maximum secretion, so guess the maximum secretion from all this, the gastric secretions from our stomach. So you think GI system means mainly, you think in your mind immediately stomach, right? So that's why you can remember like that. The maximum secretion is from stomach, the gastric secretions, that is like about 2.5 liter per day. And among this, the most acidic is also of course gastric secretions, because in the stomach the HCl is produced from our parietal cells. So the most acidic one is also gastric secretion. That's pH is important to remember. Its pH is very low because it's highly acidic, like pH is like 1.5 to 3, means the value may be in between 1.5 to 3. So the most alkaline among them is a bicarbonate secretion, which is especially coming from Brunner's gland secretion. Its pH is like 8.9. Brunner's gland, of course, it's in the duodenum, right? Brunner's gland, it's nothing but the small intestine secretion. Its pH is 8.9. And the next coming most bicarbonate alkaline solution is pancreatic secretion from pancreas. Its pH is 8.3. So once again let us revise. The maximum secretion from the GI system is coming from gastric secretions from the stomach, and the most acidic is also from the gastric secretions. The most alkaline is coming from a Brunner's gland, and next it is coming from a pancreatic secretions. Now let us start with our saliva from the beginning. So saliva, it's like producing 1.5 liter per day. And the pH is 7, that is hypotonic. The source of saliva is mainly from salivary glands, and the salivary glands are divided into major salivary glands and minor salivary glands. Your major salivary glands are the parotid glands, which are beside your ears, and you have the submandibular glands below your mandible and sublingual glands below your tongue. So these three are the major glands. Among them again the major one is a submandibular, means mostly our saliva in your mouth is coming from a submandibular gland, 70%, and next comes of course from a parotid gland, and the least one is a sublingual gland. And you have some other minor glands like von Ebner's gland. So now let us see the functions, what the saliva does. Saliva is also useful for digestion, mainly carbohydrates, lipids, but not proteins. So to remember that, I have a link like if you are eating paneer or any protein, very much protein rich cottage cheese, you may feel it tasteless because our saliva is not digesting it. So remember, in your mouth, saliva will not digest the proteins. It can digest only the carbohydrates and lipids, so that you can get the taste. So carbohydrates mainly by use of amylase, salivary amylase, which is also called tialin. And lipid is mainly by lingual lipase, which is coming from the von Ebner's gland, which is a minor gland just now we have seen. So the minor gland, von Ebner's gland, gives rise to lingual lipase, which is mainly useful for digestion of lipids. So remember, saliva can digest only the carbohydrates and lipids, but not the protein. So for protein, there is no enzymes in saliva for protein digestion. Now, and other functions of saliva, of course, lubrication. The food you eat can easily pass down into the stomach, and protection. Protection mainly by producing secretory IgA, immunoglobulin A, for the protection. And you have the bactericidal properties by lactoferrin and also lysozymes. These both are antibacterial, lactoferrin, lysozymes. LL, both L's. Now let us see the regulation of saliva secretion, how it is regulated. The regulation is completely under the control of autonomic nervous system. Autonomic nervous system is divided into parasympathetic and sympathetic. But it's a unique nature in our saliva that both parasympathetic and sympathetic here lead to increase in saliva secretion. Normally, all over the body we will see that the parasympathetic and sympathetic work in an opposite way, but only here in the saliva they both act in the same way. They both try to increase the salivary secretion. So mainly the parasympathetic is useful to make the saliva thin, watery, whereas the sympathetic tries to keep the saliva thick. So this both in turn lead to increase in saliva secretion. And cranial nerve 9 gives impulse stimulus to aortic ganglion, and this in turn activates the parotid gland, whereas cranial nerve 7 activates the submandibular ganglion, which of course activates both submandibular and sublingual glands. Means this glands which are useful to produce saliva are activated by cranial nerve 9 and cranial nerve 7. Whereas a glossopharyngeal nerve and a facial nerve. Now let us see the gastric. So till now we have completed all our salivary secretions about saliva. So from the saliva now we will move on to the stomach, which has mainly gastric secretions. So gastric secretions are maximum secretions like about 2.5 liters per day. So remember, saliva is like 1.5 liter per day, whereas gastric secretions are 2.5 liters. We already discussed the most maximum secretion is from gastric secretion, that is 2.5 liter per day, and it's highly acidic, so pH will be like about 1.5 to 3. The source from where it comes, the cells in flask shaped gastric gland. So mainly the sources, they have the flask shaped gastric gland. As we seen in saliva we have submandibular, sublingual and our parotid. Here we have the gastric gland which is flask shaped. So mainly in this flask-shaped gastric glands we have mainly 6 types of cells which are producing different types of their substances. Let us see what are those. So first we have the surface cells. Surface cells, they produce a bicarbonate rich mucus, and they are mainly useful for acid neutralization, and also they produce trefoil peptides which are having some immune function. So remember, first coming to gastric gland, you have the surface. On the surface you have the surface cells of the flask shaped cells. So that surface cells are mainly secreting bicarbonate rich mucus. Along with that they also produce trefoil peptides for immune function. And from the surface you come deeper, you have neck. For the neck they produce stem cells for stomach regeneration and found in neck. Next you have the parietal cells which mainly produce HCl and also intrinsic factor of Casel. Intrinsic factor is useful for B12 absorption. B12 is called extrinsic factor, and the intrinsic factor is produced from our stomach. So if both are present, then only the B12 is absorbed from our stomach. And if they are not present, if the parietal cell fail to produce intrinsic factor, then it may lead to pernicious anemia. Pernicious anemia is a condition where autoantibodies are formed against the parietal cell. So if our body only forms the antibodies against these cells, then they may fail to work. So this may lead to pernicious anemia. So here in this condition, it may lead to achlorhydria or also lead to anemia. Achlorhydria means there is no production of HCl, because as the autoantibodies are formed now against the parietal cells, so the parietal cells will not form either HCl or also intrinsic factor of Casel. So because of this, it may lead to achlorhydria and also anemia. Now let us see the enterochromaffin-like cells. These are also called as ECL cells. So these ECL cells mainly produce histamine. Histamine is useful to increase the acid secretion, and the drugs against histamine, you can see H2 blockers. We use ranitidine and emetidine, which are used to treat the peptic ulcer. Why we block here histamine? Because histamine is a pro-factor which is using to increase the acid secretion. So if we block this H2 histamine blockers, then like ranitidine or emetidine, we can prevent the peptic ulcer because the acid secretion decreases. And next coming to the chief cells. So chief cells mainly produce a pepsinogen. Always remember gen. At the end, if we have gen, they are inactive, so we need to convert them into active. It is converted into active only in the presence of acidic medium. So chief cells produce pepsinogen. Next coming to the G cells. G cells are seen particularly in antral region of the stomach. Already we know the parts of the stomach. Antrum is like the base of the stomach. So there the G cells mainly produce gastrin. G for G cells produce a gastrin hormone. And now let us see the parietal cells. The parietal cells produce a gastric acid HCl. For every HCl produced, the bicarbonate ion enters the blood vessel. Actually, this is a diagram. What happens in parietal cells, the mechanism why the parietal cells forms the HCl. Let us see. So actually here you can see mainly HCl is produced by our parietal cells. For every HCl produced, bicarbonate ion enters the cell. So here you can see as the HCl is produced, the bicarbonate is entering into the blood vessel. So postprandial alkaline tide, this is called as postprandial alkaline tide. Once I will explain this mechanism. Actually what happens is, here we have a pump called as hydrogen potassium pump. Actually first this process begins with carbon dioxide. You know, carbon dioxide, when it reacts with water in the presence of carbonic anhydrase, it forms a carbonic acid. This carbonic acid in turn breaks down into the bicarbonate and H plus ion in the basolateral parietal cell. So this H plus is pumped out of the cell into the lumen. As the H plus is coming out, the proton pump, in the presence of ATP, it brings the potassium ion into the cell. So because of this, H plus has come out. Now we need Cl for formation of HCl, right? So the chlorine comes out here, means this bicarbonate goes into the blood vessel, so that chlorine comes from the blood vessel directly into the lumen. So this is how the HCl is produced. So you can understand here the mechanism. Whenever the HCl is produced into the lumen, the bicarbonate, the alkaline nature is increasing in the blood vessel. So whenever you can find out, whenever the acid is produced from the stomach, always the alkaline nature in the blood increases. So that is called as postprandial alkaline. Postprandial means after eating. After eating, there is alkaline tide in a blood vessel. So increase acid production after food take and increase bicarbonate ions in the blood. Now here let us see the agents which are altering the gastric acid secretion. So mainly whenever there is higher proton pump activity, it will lead to higher acid secretion. Because here you can see in this diagram, higher proton pump activity means more hydrogen ions comes out into the lumen and more potassium goes into the lumen. So because of this, there is higher production of HCl. So what are the factors which try to increase the proton pump activity? Those are like most potent histamine through H2 receptor on the parietal cell. So remember, histamine have H2 receptor on the parietal cell, and gastrin through cholecystokinin, that is CCKB receptor, and acetylcholine through muscarinic receptor, especially M3 receptors. So remember, histamine, gastrin, acetylcholine. These are the three important factors which increase the acid secretion. So there are other factors which decrease acid secretion, means they are decreasing the proton pump activity. So what are those? We have the gastric acid, which is negative feedback. It inhibits the further production. So whenever there is higher gastric acid in the stomach, then it tries to decrease the gastric acid by a negative feedback inhibition. And we also have somatostatin. In the name itself, this statin, something is blocking, so it's a universal inhibitor. And also you have a prostaglandin, that is PG blockers. So actually what the prostaglandin does, it decreases the acid secretion. So actually we use the prostaglandin blockers like NSAIDs, which cause a peptic ulcers as side effect. Why they cause peptic ulcers? Because they are blocking the PG. PG blockers are mainly decreasing the acid secretion. So if you block the prostaglandins, then there is more acid secretion. If there is more acid secretion, so because of using this NSAIDs, you get a peptic ulcer as a side effect. Now let us see the role of gastric secretion in digestion. So now till now we have seen like in mouth there is no protein in digestion. Now let us find out in stomach whether all substances are in the food are getting digested or not. So lipids in stomach are they're digested by means of gastric lipase. They have an enzyme gastric lipase, and proteins in our stomach are digested by pepsinogen. Actually it's inactive. Whenever there is HCl, it converts into active pepsin. So this pepsin digests the proteins. And we also have renin, which is particularly found in cows, you can remember it, and carbohydrates, no enzymes in stomach. So exceptions are important. In the mouth there is no digestion for proteins. In the stomach there is no digestion for carbohydrates. But there are some interesting phases going on in gastric secretion. What are those? First one is cephalic phase, second one is gastric phase, major phase is a gastric phase, and the third one is an intestinal phase. So cephalic phase is just by seeing the food, just by thinking the food, or just by taking talking about the food, but not intaking food. Means just you are thinking or talking about the food, just by that only like 20% of gastric acid is always secreted. And next, coming to gastric phase major, it is like food when reaches the stomach. When the food actually you are eating the food, then the major secretion is like 60 to 70% of acid secretion, and intestinal phase is 10% of acid secretion. So let us complete it. So we have completed about stomach. Now from the stomach, let us go into small intestine, because exocrine pancreatic secretions are finally released into small intestine. So let us see what happens here. The alkaline pH of this pancreatic secretions are 8.3. They are bicarbonate rich. We have seen the major bicarbonate rich is our Brunel's gland secretion, and next comes the pancreatic exocrine pancreatic secretion. So why we are stressing here exocrine? Because endocrine pancreas is mainly useful for production of insulin, right? Those all hormones. Here it is mainly producing enzymes for digestion. So here the carbohydrates are digested by pancreatic amylase and lipids by pancreatic lipase major, and also it has as helper colipase, and the proteins by means of trypsinogen, chymotrypsinogen, procarboxy peptidase. So all these are this procarboxy peptidase is a zymogen. All these are inactive proteins. They are activated and they are useful for protein digestive enzyme. And here this zymogens means protein digestive enzyme are stored in inactive form to prevent autogenation of pancreas. So actually we get doubt why they are inactive, because if they are active they may even digest away the pancreas. So to prevent that digestion, we put them in inactive form and later they are converted into the active form. So you may get a clear idea till now that in mouth by saliva there is no protein digestion, in stomach there is no carbohydrate digestion, but by pancreatic secretion all are digested. You can see carbohydrates are digested, lipids are digested, and also proteins are digested. Now let us move on to small intestine. So in small intestine the trypsinogen is inactive. It is converted into active form by trypsin by means of enterokinase and removal of few amino acids. So the regulation of pancreatic secretion is done by hormonal. It has mainly two hormones for regulation, that is CCK-PZ, that is Cholecystokinin Pancreaozymine, and also Secretin. So let us see how these are regulating the pancreas. So PZ is pancreatic secretion, which is rich in digestive enzymes. So pancreatic secretion rich in digestive enzymes. CCK is useful for contraction of gallbladder to release of bile. So CCK in the name cholecystokinin. Cyst means gallbladder. So remember, cholecystokinin used for contraction of gallbladder. By this it leads to release of bile. And secretin is poor in digestive enzymes. It's alkaline, rich in bicarbonate and acid neutralization. So secretin is major component of pancreatic juice, which is leading to alkaline nature because of having more amount of bicarbonate. So by this we completed the pancreas. Now we will move to biliary secretion, that is bile from the liver. So produced by liver, so called as liver bile. So remember, it's like 500 milliliter per day, means 97% of water content and stored and concentration in gallbladder. So then it is called gallbladder bile. It is 87% water content. So that is why we are saying it is concentrated in gallbladder, because the water content is decreased. Whereas in liver you can say it is 97% water content, but in bile, gallbladder bile, it is 87% water content, because most of it is concentrated and reabsorbed by the gallbladder. Now what are the constituents of bile? Bile have mainly bile acids, it have bile salts, it have biliary pigments. So let us see what are bile acids. Bile acids are cholic acids, and it is also called as coniodeoxycholic acid. And the primary bile acids, and in the presence of intestinal bacteria, they are converted into the secondary bile acids. These secondary bile acids are called deoxycholic acid and also called as lithocholic acid. So remember, the bile acids are cholic acids, coniodeoxycholic acid, and these are the primary bile acids. In the presence of intestinal bacteria, they are converted into secondary bile acids. So the conjugation of bile acids takes place in the liver when they combine with glycine or taurine. So when a bile acid combine with glycine or taurine, they form a conjugated bile acids. So next, let us see the bile salts. So the salts of sodium and potassium are the bile salts. Examples: sodium glycocholic acid and bile pigments and cholesterol. So these are all the components of a bile. Now let us see the function and regulation of bile. The function of bile is for digestion and absorption of exclusively lipids in diet. So mainly bile is used to digest the lipids and excretion of cholesterol and bile pigment. So regulation is done by coagules and colectrins. Coagules are contraction of gallbladder and release of bile, that is cholecystokinin, and colectrins are which increase the bile secretion. And bile salts are most potent and secretin and vagal stimulation. These are all the colectrins which increase the bile secretion. And coagules are which lead to contraction of gallbladder. So of course we have a cholecystokinin and coagules, and in colectrins we have bile salts, most potent one, and secretin and vagal stimulation. Now let us see the intestinal secretion. So intestinal secretion are mainly released from crypts of lubricants, so especially from the globular cells or surface cells, which produce a bicarbonate rich mucus and also useful for acid neutralization of the food which is coming from the stomach. So we also have epithelial cells which are intestinal secretion called as succus entericus, and Pennant cells which are present in the base. They are high concentration of zinc, and they produce the defensins and lysozymes which are both antibacterial. So these are the intestinal secretion. Now we have to see the potassium secretion. Potassium secretion is maximum potassium secretion is milliequivalents per day is from saliva. S for S, maximum secretion of potassium maximum in saliva. Highest potassium concentration milliequivalents per liter. Concentration C for C, colonic secretion, or also called as rectal fluid. So it has maximum potassium concentration. It has saliva as maximum potassium secretion milliequivalents per day. It's milliequivalents per liter, colonic secretion, rectal fluids. So what are the layers of GIT? From outside to inside. Outside we have serosa, inside we have muscularis propria, which also have mesenteric and orbic plexus, which regulate the GI motility pattern. And we have the inner submucosa. Submucosa have the mucinous plexus, which regulate the GI secretions. And so mucosa, we have muscularis mucosa, which alter the surface available for digestion and absorption. There is no role in GI motility. So by this, we completed our GI secretions. And next we'll continue our GI hormones. Let us continue the gastrointestinal hormones. So there are two types of families in gastrointestinal hormones. It is a gastrin family and secretin family, and also we have some other types of hormones. So in the gastrin family, mainly we have a gastrin and cholecystokinin, and in a secretin family we have secretin, vasoactointestinal peptide, that is VIP, gastric inhibitory peptide, that is GIP, and glucagon-like peptide GLP. So in gastrin family, gastrin and CCK. In secretin family, secretin, VIP, GIP and GLP. Next, coming to other type of hormones, we have motilin, guanil, peptide YY and ghrelin. So these are all different types of hormones. So now let us see the sources and hormones, means from which type of cells these hormones are released. So from G cells, G for G, from G cells gastrin, S cells secretin, I cells cholecystokinin. You can see here in the kinin word the I is repeating 2 times, so you can remember like that. From I cells cholecystokinin, and from delta cells somatostatin, MO cells motilin, motilin, K cells gastric inhibitory peptide. So here the different ones to remember are delta cells somatostatin, K cells GIP, all others are their starting letters. So next we'll see the gastrin in detail. So gastrin is produced from the G cells. This G cells, S cells, all these source cells are called enteroendocrine cells, and these G cells are present in the antrum. From this antrum this gastrin is produced. So it's the most important gastrin which is in circulatory form is G17. And the factors that increase the gastrin levels, the most important one is a peptide and amino acids. So if there are more, then it increases the gastrin levels. And antral or any antral distension of food in antral region, if there is distension by means of food or any other substance, or also gastrin releasing peptide also may increase the gastrin levels. Actually, this gastrin releasing peptide is released by our parasympathetic vagal nerve endings, or whenever there is calcium or epinephrine. So this may also lead to increase in the gastrin levels. Now let us see what are the factors which decrease the gastrin levels. So excess acid tries to inhibit the G cells, so in turn also decreasing gastrin levels. And somatostatin, it's a universal inhibitor, and also gastric inhibitory peptide. In the name itself you can see it's inhibitory peptide, and also secretin. So remember, these four are the inhibitory means which are decreasing the gastrin levels. And the proteins, amino acids, distensin, gastrin, releasing peptide, calcium, epinephrine, these are all the increasing factors. So the function of gastrin are due to a receptor. So it should act on a receptor, that is cholecystokinin B receptor. So remember, gastrin acts on cholecystokinin B receptor, and the functions include to increase acid secretion to parietal cells. Of course, by the receptor it activates so that there is increase in acid secretion from parietal cells, and also there is increase in pepsin. There is also increase in pepsin secretion, which aids in protein digestion. You know, pepsin is used for protein digestion in stomach. And also there is increase in GIT mucosal growth, that is a tropic action, means there is growth in the mucosa cells of GIT. So now let us see the Cholecystokinin Pancreaozymine. So this is also belonging to our gastrin family, right? So this is produced by eye cells in intestine, and also present in GIT and brain. So remember, cholecystokinin pancreaozymine is also present in brain. We know already it's present in GIT, but the rare thing is it's also present in brain. So what are these functions? So mainly the CCK is useful for contraction of gallbladder. So and also it relaxes the sphincter of Oddi. So the main function to contract the gallbladder, or either to relax the sphincter of Oddi, is mainly to release the bile, because if gallbladder contracts, bile release, and even the sphincter of Oddi relax, the bile can easily flow out. And PJ, that is a pancreaozymine, makes the pancreatic secretion rich in digestion of enzymes. So you know, as the pancreatic secretion have more enzymes, so that it can easily digest. And CCK augments the action of secretin, means CCK try to increase the action of secretin, because both are like inhibitory, right? So they both can support each other. And it also inhibits the gastric emptying, because there should be sufficient time for the small intestine to act upon. And in brain, satiety means decrease in food intake. So in brain it tries to decrease in food intake, and in anxiety, in analgesia, all this. And CCK is a gastrin family. It's also having a trophic action which stimulate the growth of GIT. So before you see increase the GIT mucosa growth, even here CCK also stimulates the growth of GIT. And CCK actions are on CCK-A receptor. So before we have seen gastrin is acting on CCK-B receptor, whereas this cholecystokinin is acting on A receptor. And the major stimuli for the CCK secretion is either peptides or amino acids in that, the same which we have seen in the gastrin. So now let us see the secretin. So we are coming to the next family. We are completed the gastrin family. You know, in secretin family, you know, we have secretin, GIP, VIP, GLP, right? So let us see the secretin. Secretin is actually a first hormone which is discovered in humans by Baylis and Starling. So among all hormones, secretin is the first one, lucky hormone to find out one. So it is produced by S cells, and the functions of secretin are: the pancreatic secretion is rich in bicarbonate. So it tries to make the secretions rich in bicarbonate ions, because actually it's stimulated whenever the chyme is more acidic. You know, as the acidic chyme enters the small intestine, it's get activated and tries to produce more bicarbonate ions, because now it want to neutralize that acidic chyme. So therefore it is also called as nature's antacid. Antacid means what? It tries to neutralize the acidic nature. So it is called nature's antacid, because it's naturally producing the bicarbonate whenever there is acid chyme entering the small intestine. And it also augments the actions of CCK. Augments means it also tries to increase the action of cholecystokinin, because both you can see both are having some inhibitory function, both are inhibiting the acid secretion, right? So both can each other increase their action. And stomach actually decreases the gastrin, because you know, in stomach there is more acid production, and decreases the gastric acid, yeah. and increase contraction of pyloric sphincter. Actually, secretin does these things in stomach. Secretin decreases the gastrin in stomach, and secretin also decreases the gastric acid production, and secretin increases the contraction of pyloric sphincter in the stomach. Now let us see the VIP, GIP and GLP. So VIP, vasoactive intestinal peptide. It's mainly a relaxing agent in GIT. It increases the fluid and electrolyte secretion. In blood vessels, it's lead to vasodilator because of dilating the smooth muscles in blood vessels and also in GIT. So it's like a relaxing factor. And next, coming to gastric inhibitory peptide GIP and glucagon like peptides GIP-AR. So these are like GI hormones which increase the insulin secretion. So as they are increasing the insulin secretion, they are called incretins. And we have the GIP, which is a glucose dependent insulin-trophic peptide. Next, coming to motilin. So we have completed the family of gastrin, secretin. Now we are going to see the other hormones remaining like motilin, guanine, peptide YY, ghrelin. So let's start with motilin. So motilin is produced by most cells and they increase the GI tract motility called MMC by motilin receptor. So erythromycin actually it stimulates this motilin receptors. So the major side effects is diarrhea. More motility means of course more excretion of fecus. So erythromycin gives a side effect of diarrhea by acting on this motilin receptor. So increased GI motility used for the treatment of GI motility disorders. Example, gastroparesis. Gastroparesis is seen in diabetic patients who have low GI motility. So to improve their GI motility, we can suggest we can give erythromycin as a treatment so that it acts on the motilin receptor and increase the motility. And next, coming to guaniline and peptide YY. So guanilin increases the chloride ion secretion in GIT. So more chloride ion secretion by guanilin, and E. coli has a structure which is similar to guanilin. So sometimes E. coli may go and act on this receptor and it may mimic it, right? So can activate the guanilin receptor and increase in chlorine secretion. Even E. coli tries to increase the chlorine secretion. So this is called as molecular mimicry. Means instead of guanilin, now the E. coli will go and act on the receptor and try to increase the chlorine secretion. Next, let us see the peptide YY. So peptide YY is a major stimulus. Its major stimulus is fat. So whenever there are fats, it gets activated and it inhibits the intestinal motility at terminal ileum. Ileal break phenomenon. This is called ileal break phenomenon because it's terminating the motility of ileum. Next, coming to ghrelin. Ghrelin is produced in stomach, in oxyntic gland region, and the major functions of ghrelin is increase the food intake. It is also called as hormone of hunger because we are eating more food, means it's because of hunger. Especially when you are in fasting for longer time, then you'll have more ghrelin, of course, because you are fasting, so more hunger. And growth hormone, it also leads to growth hormone secretion and also leads to increased insulin secretion. So actually in the name you can say GHRE, GH, growth hormone release, like that. And INS, increase in insulin secretion. And also in stomach you can see increased gastric motility, increased gastric acid secretion of more motility, more secretion in stomach by ghrelin. So by this we completed our GI hormones. Next we'll continue our digestion and absorption. Digestion and absorption. So first we have the carbohydrate digestion. So for the food we eat, in that the carbohydrate, the source is like starch. It's a complex polysaccharide. So now this complex polysaccharide need to be converted into a simpler form to get absorbed into our body. So mainly starting from saliva, the saliva has salivary amylase, which is it can digest only 5% of carbohydrate. It is mainly activated by the chlorine ions which is present in the mouth. But why only 5% of carbohydrate is digested by salivary amylase? Because it immediately gets deactivated by acid in the stomach. So only from the mouth 5% of carbohydrate digestion is done. And coming to stomach, we already discussed before, in stomach there is no enzyme for digestion of carbohydrate. So no digestion of carbohydrate in stomach. Next, the pancreas contains pancreatic amylase, which is a major important one which leads to 95% carbohydrate digestion, which acts at the level of 1:4 alpha linkage. So because of this, this polysaccharide is now converted into disaccharide or trisaccharide, and this disaccharide or trisaccharide in the presence of lactase, sucrase and isomaltase, which are present in a mucosal surface of level of 1:6 alpha linkage of a small intestine. So next they are converted into monosaccharides. The monosaccharides are nothing but the glucose and fructose. So now this can easily get absorbed into our body. So now let us see the intestinal glucose and fructose absorption. So till now we understood the digestion of carbohydrate. So now let us see how this carbohydrate is now absorbed into our blood vessel. So first you can see always sodium and glucose ion are co-transported, means they both go inside the cell into the intestinal cell. This is a apical cell, this is a lumen, this is a basolateral end. So now this both are going by means of sodium glucose co-transporter, which is also called as SGLT1 in intestine, and this is a secondary active transport. So now this both go together inside, and after that by means of GLUT2, that is a facilitated diffusion, now the glucose goes from the basolateral side into the blood vessel. So this is a simple transport by mainly sodium glucose co-transporter and GLUT2. So this to facilitate the glucose transport from the lumen into the blood vessel. So glucose is transported into intestinal cell from the lumen by SGLT1 along with sodium, and this is why it is also used in ORS solutions. Oral rehydration solutions also contain, that's why sodium and glucose they both can easily get transported by means of this co-transporter. There is a secondary active transport. So from the intestinal cell into the blood vessel, the glucose is transported by GLUT2 transporter in a basolateral region, that is by facilitated diffusion. So the fructose absorption is transported by GLUT5, that is facilitated diffusion. So you can see, as we said, glucose and fructose both are monosaccharides. Glucose have this kind of pathway, whereas fructose also follows the same, but the only difference is in the fructose it uses, instead of here GLUT2, it uses GLUT5. How to remember it uses GLUT5? 5 starts with F, and fructose also 5, and F again for facilitated diffusion. So GLUT5 instead of GLUT2 in the case of fructose. Now let us see the lipid digestion. So now we understood about carbohydrates, how it gets digested, absorbed. Now let us see how lipids get digested and absorbed. So mainly lipids in our diet are in the form of triglycerides, and in mouth, in the saliva we have lingual lipase, in the stomach we have gastric lipase, and in a pancreas we have pancreatic lipase. So remember, for lipid we have everywhere the enzymes, and in pancreas we also have the colipase and cholesterol esterase. So these are all the enzymes which are required. So now this triglycerides in the presence of lipase, colipase and cholesterol esterase in the pancreas plays a major role. Mouth and stomach only somewhat role. Pancreas is again a major role. Even in our glucose and fructose transporter also pancreas is playing a major role. So now here these triglycerides are converted into monoglycerides, which are nothing but free fatty acids, cholesterol, fat soluble vitamins like A, D, E and K. So these are all now incorporated into a small micelle. This micelle is a transported vehicle. So high concentration of free fatty acids are present in the micelles, and this is transported. So the micelles come closer to the mucosal cell, and the free fatty acid move into the mucosal cell. Because already in the mucosal cell there is no free fatty acids. So now the free fatty acid, according to concentration gradient, just by passive diffusion, it moves from the lumen side into the mucosal cell. So now in the mucosal cell the free fatty acids are again re-esterified into triglycerides. Why again they are re-esterified? Because they need to keep always the mucosal cell free fatty acids low. Then only again next free fatty acid can come into the cell by means of passive diffusion. So after that, these triglycerides are now packed in the form of chylomicrons, and these are formed by our triglycerides, and these are now transported into whole of our body. So this is how the fatty acids get digested and get absorbed into our body. Now let us see the bile. So in bile, mainly the bile salts from the liver are going to the intestine, especially to the terminal ileum of the intestine. They get absorbed there, and after that again by means of enterohepatic circulation they again go into the bile salts. So why this process is going on? Why the new bile salt is not forming? Why only recycling? See, First from the bile salt into the intestine, again from the intestine into bile salt. Hence the name enterohepatic circulation, also called as recycling of bile salt. So the bile synthesis is actually energy dependent to reduce the demand to form new bile salts. So actually this process, this recycling is going on because this is actually highly energy dependent. So to reduce that energy, we are trying to recycle it many times. And the recycling of bile salts like this per day, it takes like about 6 to 8 times per day. Means that many times the bile salts are getting absorbed into intestine, and again they are recycled. Now let us see what are these short chain fatty acids, in short form SCFA. So these are mainly produced by bacteria in the colon, and they are like propionate, butyrate and acetate. So three types of short chain fatty acids are propionate, butyrate and acetate. So their functions are mainly they have trophic action, means they try to increase the mucosal growth, and also they have the acid base balance during absorption. The hydrogen ionized exchange, means when they are absorbing anything, the hydrogen ions exchange so that there is acid base balance, and also they try to prevent the inflammation. So these are the main functions of the propionate, butyrate and acetate, which are the short chain fatty acids produced especially in a colon by means of bacteria. Next we will move on to the protein digestion and absorption. So protein digestion in the diet mainly it is present in the form of polypeptides, and in the saliva, remember, there are no enzymes for the protein digestion, whereas stomach we have pepsin, and in pancreas, of course, you can see in any kind of digestion our pancreas are playing major role for the digesting most of them. So in the pancreas again we have trypsin, chymotrypsin, which are mainly endopeptidases, means they break down the peptide bonds mainly in the interior region, so they are called as endopeptidases, trypsin, chymotrypsin. And coming to carboxy peptidases and aminopeptidases, these are exopeptidase, means they act on the bonds in the end region, means they break down the branch on the end region. So the enzymes from pancreas are actually in inactive stage because they get activated only in small intestine, because otherwise it may lead to autolysis of pancreas itself. So proteins in the presence of peptidases, they convert into dipeptide and tripeptide, and this dipeptide and tripeptide, you can see in this diagram, they are transported by means of PPT. PPT is a transporter, peptide transporter, which transports the dipeptide, tripeptide and also hydrogen ion into the intestinal cell from the lumen. So this H+ is transferred out by exchange with sodium ion using the sodium hydrogen exchanger. So you can see as a hydrogen ion has come inside, now it is getting out, so that sodium is getting in by means of sodium hydrogen exchanger. So now the dipeptides and tripeptides inside the cell are broken down to amino acids by means of cytoplasmic proteases into amino acids. So now these amino acids are transported into the blood vessel by basolateral amino acid transporter. So now the amino acid is finally come to blood vessel. So proteins are absorbed in our blood vessel by means of amino acid. So first we need to convert the dipeptide, tripeptide in the cell into the amino acid by means of cytoplasmic proteins. So now let us see the vitamin absorption. So vitamin absorption, we have two types of vitamins, which are fat soluble and water soluble. For fat soluble vitamins like A, D, E, K are carried in the form of micelles. We already discussed in fat digestion. There it is absorbed into the mucosal cell. Whereas in the water soluble, they are absorbed by sodium dependent co-transporters, except folate and vitamin B12, because they are sodium independent. Like folate is absorbed in jejunum, and vitamin B12 is absorbed in the ileum. They are sodium independent. So the GIT has daily total secretion of like 9 liters, and the maximum of water absorption in the GIT is done by the jejunum. Now let us go on to the iron absorption. So iron absorption is mainly is done in the duodenum, that is in the upper part of small intestine. In the diet it is present mainly as ferric form, that is Fe+3 form, and its absorbable form is Fe+2 form. So Fe+3 is converted into Fe+2 by means of ferric ductase form, and now this is transported into the cell by means of a bivalent metal transport. By means of a divalent metal transporter, that is called as DMT1. So now this DMT1 is converted into Fe+2. So first again let us repeat, the major site is in duodenum. So it is present in ferric form, means Fe+3 form, but absorbable form is Fe+2 form. So now we need to convert this into Fe+2. How we will convert? By means of an enzyme, that is ferric ductase. So now this Fe+2 is transported into cell by divalent transporter. So now this Fe+2 is present in inside a cell. For example, this is cell. So inside the cell now we have Fe+2. So Fe+2 is also formed by another mechanism like heme. Heme which is present, it is heme carrier protein. We have a heme carrier protein which also converts the heme into the intestinal cell. This also converts now into Fe+2. So now Fe+2 is coming from two sources. One is from heme and other is from directly from Fe+3 converting into Fe+2. So now this both Fe+2 which are present in the cell need to transport it to the blood vessel. How they will transport? So now this both Fe+2 are converted into again Fe+3 and stored as ferritin. So the remaining Fe+2 is transported out by basolateral end by ferro portion. So actually, for example, you think this is a cell in which now the iron is getting absorbed. So we are having two sources for iron absorb. One is coming from heme and the other is coming from the direct Fe+3. So this both are converting into Fe+2. Now again this Fe+2 inside the cell is converting into Fe+3 and called as ferritin. And that ferritin, okay, it is formed, but remaining some more Fe+2 is remaining. That remaining Fe+2 is transported by means of a ferroportin. You can see here. Where is that? By ferroportin. So that ferroportin is converted to Fe+3 by hepastine. So whatever, again that ferroportin is also converted into Fe+3 by hepastine, and this Fe+3 moves into the blood vessel and it is transported with transferrin. So this is how it is again converting into first Fe+2 into Fe+3, that is called ferritin, that is stored. Okay, leave it that. Now that remaining Fe+2 is transported by means of ferroportin, and that ferroportin again it is converted into Fe+3 by hepastine, and again that Fe+3 moves into the blood vessel and it is transported with ferritin. So finally Fe+3 is a transportable form in the blood vessel. So factors that increase the iron absorption are vitamin C. So all this process, vitamin C can increase, that is ascorbic acid of course. And the factors that decrease the iron absorption are tannins which are present in tea, phytalates and oxalates. Actually, they form complexes with ferrum so that they prevent the iron absorption. And regulation of iron absorption is done by hepcidin. Hepcidin is released from the liver. So actually what hepcidin does is it decreases the iron absorption. How? That mainly by acting internalization and destruction of ferroportin. So you can understand now the ferroportin is the one which need to transport from the basolateral side into the blood vessel. So if that ferroportin is not there, it cannot transport, so thereby decreasing the iron absorption. So remember, hepcidin is a negative factor, it is decreasing the iron absorption. So hepcidin mutation leads to iron overload. Yes, of course, because hepcidin is the one which it's like a regulatory process. Whenever it's more, we need to destruct it. But here there is no hepcidin. If there is no hepcidin, then more iron will get absorbed into our body. That is called as hemochromatosis. So by this we completed our quick revision of our digestion and absorption, and next we will continue with our last part, GI motility. Channel, let us continue the GI motility. So mainly the movement of food is done by the smooth muscles which are involuntary, means not in our control. So the food movement is involuntary. So as it is involuntary, it has gap junctions and also pacemaker. Always any smooth muscle is involuntary, it will have a pacemaker. So here the gap junctions are where the smooth muscle act as a single unit, and the pacemaker is here the interstitial cells of Kachal. Now let us see the restless. Whenever a cell is involuntary and it has a pacemaker, then of course it will have a restless membrane. Pacemakers always have a restless membrane potential but not resting membrane potential. We already discussed this in a cardiac physiology. So the restless membrane potential is found mainly in Kachal cells. So where the resting membrane potential is ranging between -60 to -40. And the basic electrical rhythm is formed here, which is also called as slow waves. So these are actually oscillating. The resting membrane potential means they're just oscillating like this between the resting membrane potential and threshold, but they will not go beyond threshold. If they are going beyond threshold, then they will form the spike potentials or action potentials. So as I said, these are just between the resting membrane potential and threshold. They are not true action potentials. As they are not true action potentials, they also don't have any role in muscle contraction. And this frequency, the maximum frequency of this basic electrical rhythms are seen in duodenum, which is 12 per minute, and in ileum it is 8 per minute, whereas minimum it is seen in cecum, that is 2 per minute. So the action potential of a smooth muscle mainly consists of depolarization and repolarization, like all muscles. So the firing level for the action potential is -40, because as we said, it is resting membrane potential is -60, the threshold is -40. So if the this basic electrical rhythms reach -40, then they start producing the action potentials. So the firing level for action potential is -40, and the depolarization is due to mainly calcium influx here, and the repolarization is same like our skeletal muscle or normal, that is our potassium efflux. It's a universal that the repolarization is due to potassium efflux, and the depolarization is mainly due to calcium influx. So the interstitial cells of Kajal are pacemaker cells and exclusively located in the stomach region, that is especially in the mid body along the greater curvature. So in the greater curvature stomach region, mainly this interstitial cells of Kajal pacemakers are located. Now let us see the motility patterns. So the first motility patterns, whenever you eat the food, is the deglutition. Deglutition means nothing but the motility is the deglutition. Deglutition is nothing but also called as the swallowing of food. So in the swallowing of food, we have the three phases. The first one is the oral phase, and the second one is the pharyngeal phase, and the third one is the esophageal phase. So the oral phase means in the mouth we do, it's voluntary, you can control it, it's voluntary. Whereas the pharyngeal and esophageal, it's involuntary, you can't move them, right? So these are involuntary. So first one is a deglutition. So there is a reflex for deglutition. How it is first efferents. Efferents are coming from cranial nerve 5, trigeminal nerve 9, glossopharyngeal and 10 vagus. So these three nerves, trigeminal, glossopharyngeal and vagus, these are the efferents. They give the efferent to the central medulla. So central medulla, especially the location is in the nucleus rectus solitarius. So from this nucleus rectus solitarius, the efferents will come. The efferents here are again the trigeminal and the facial and the hypoglossal nerve. So these three will go to the muscles of pharynx and tongue. So by this they are concerned with swallowing. So this is how the muscles of pharynx and tongue are activated. Now next let us see the esophageal motility. So the esophagus motility is most important movement for peristalsis, that is peristaltic contraction. So this peristaltic contraction are divided into three types: the primary peristalsis contraction, the secondary peristalsis contraction and the tertiary. The primary peristaltic contraction is called most physiological contraction because it is the primary contraction is progressive and it starts immediately on swallowing. So whenever you start swallowing food, it starts. Whereas secondary is the progressive, it is seen only in irritation of esophagus. So whenever only the esophagus is irritated, then the secondary starts. It's also progressive. But remember that territory is non-progressive, means it is like starting and stopping like that. It's intermittent. So this territory may lead to the esophageal spasms. So esophageal spasm means it will be continuously in a either contracted or relaxed phase, that is called as esophageal spasm, that is territory one. Now let us see the peristaltic movement. So peristaltic movement is mainly initiated by a local stretch of gut. Whenever the food is present, then the peristaltic movement starts. So it mainly releases the serotonin. So serotonin activates the myelinic plexus, which is present in our muscularis externa, or also called propria. So this leads to peristalsis. So this is how serotonin releases and it activates the peristalsis process. So whenever the food is in the bolus, that part gets distended and the before parts get contracted and the later parts get relaxed. Because of this contraction, the food moves front, and as it relaxes, now the food can move easily. So this is the peristalsis process. So you can see the segment before the food undergoes contraction. It is a retrograde contraction which pushes the food forward. So retrograde contraction pushes the food forward due to mainly release of acetylcholine from a parasympathetic and also substance P. So acetylcholine, substance P is nothing but substance peristalsis. So acetylcholine, substance peristalsis lead to contraction, retrograde contraction. Whereas coming to the segment after the food bolus undergoes relaxation, and this part is anterograde relaxation, it receives the food. So it is mainly due to relaxation is due to nitric oxide production and also vasoactive intestinal peptide, that is our VIP. receptive intestinal peptide SR VIP. So by this mainly, so remember the retrograde contraction is due to acetylcholine substance P, and the anterograde relaxation is due to nitric oxide and VIP. Now let us see achalasia cardia. So what is achalasia cardia? It's a peristaltic problem. So here actually there is no relaxation by means of nitric oxide. So you can see absence of nitric oxide, but acetylcholine is present. So acetylcholine is present, there is continuous contraction but no relaxation. If no relaxation also, the peristalsis movement may not raise blood, may lead to achalasia cardia. So it is unopposed, means acetylcholine is unopposed, so there is sustained contraction at the level of lower esophageal sphincter. So the treatment for this is you can give Botox, that is botulinum toxin, which can inhibit the acetylcholine release. So treatment is botulinum toxin. Next, coming to the gastric motility. So gastric motility is like receptive relaxation, or also called as gastric accommodation. So actually what is this? It is a storage of food, mainly useful for storage of food. Actually whenever the food comes, the stomach's proximal part relaxes on receiving food. So whenever the food is coming, to receive that food, the stomach will undergo relaxation and it will receive the food. That is called as receptive relaxation. What is retropulsion? Mixing and grinding of food is called retropulsion. Actually the food, when the food is traveling to the closed pylorus, the end of the stomach part, if that pylorus is closed, again the food will come back and mix. So that is called stomach repetitive cycle till the pylorus is closed. So here the mixing and grinding of food takes place. Next, let us see the gastric emptying. So gastric emptying is nothing but the stomach, the small intestine control lies in the pylorus. So mainly the stomach small intestine control lies in the pylorus. So what are the factors which increase gastric emptying and decrease gastric emptying? Let us see. The factors which mainly increase the gastric emptying are increase in gastric volume or gastric disease, that is more food is coming so that it can empty, and also due to the hormone gastrin. Whereas the factors which decrease the gastric emptying are enterogastric reflex. So what is this reflex? It is the most important enteric reflex. Actually what it does is it tries to stop the stomach from emptying. So which is inhibited by presence of certain undigested and unabsorbed substances in intestine like carbohydrates, lipids, proteins, acid, hyperosmolar content. So actually the name is enterogastric reflex. Decrease entero means small intestine cells are inhibiting the gastric system, means inhibiting the stomach emptying, means the stomach should not empty now, because already there are a lot of undigested and unabsorbed substances which are present in intestine like carbohydrates, lipids, proteins, acids, hyperosmolar content. So here the major one which can decrease gastric emptying is cholecystokinin, so it's inhibiting one, whereas this is activating one, that is gastrin. Now let us see the small intestine motility. So the small intestine motility is mainly by peristalsis, segmentation. So peristalsis is also seen before in everything in a gastric motility, but in small intestine motility the important one is segmentation. So segmentation is a process where before the food and after the food there is contraction. So in peristalsis we have said before contraction, later relaxation, but here two sides contraction. Because of two sides contraction, the food is stuck inside, so that it gets greater time for absorption and also greater time for digestion. So segmentation is contraction at both ends, retards the food movement to give adequate time for digestion and absorption. Now let us see what is large intestine motility. So large intestine motility is divided into two parts, proximal colon and distal colon. That is large intestine. So proximal colon, it's mainly for absorption of water and electrolytes, whereas distal colon is useful for storage of fecus. So the proximal colon that's it. So the maximum colonic activity at the level of sigmoid colon, very important. The maximum colonic activity is seen as sigmoid colon. Let us see the actually the movements patterns in large intestine are divided into two types, horstrations and mass movements. What is horstrations? It's a combined contraction of both circular and longitudinal. In our muscularis externa we have a lot of smooth muscles with circular and longitudinal muscle. So their combined contraction, if they contract combinedly, that is called horstration. It is exclusively seen in proximal colon, because proximal colon is absorption of water and electrolytes, so combined contraction, so more time for absorption of water and electrolytes are horstration. What are mass movements? Mass movements involves large area of colon. So mass movements in the large area of colon, everything moves from one segment to other and eventually everything is pushed towards the colon. So it's like useful for defecation process, because mass movement means at a time all are moving, not like segmented. It's completely different from segment. Segment only some will move. Here at a time everything will move, so that there is higher force for defecation reflex. So it is rectum distension, defecation reflex is initiated here. So So defecation reflex is controlled by two sphincters. So main sphincters are internal anal sphincter and external anal sphincter. So the internal anal sphincter is a smooth muscle and it's mainly involuntary, whereas external anal sphincter is a skeletal muscle and it's voluntary. So internal anal sphincter is involuntary. Involuntary means it's not in our control, it's controlled by either parasympathetic or sympathetic. So whenever parasympathetic comes, it's relaxing stage, so it facilitates defecation, whereas sympathetic is in a stressful state. So in stressful state it inhibits defecation, so in stressful state there is no chance of defecation, but in parasympathetic stage there is defecation. Let's come into external anal sphincter. So external anal sphincter is skeletal muscle voluntary, supplied by pudendal. So external anal sphincter is actually voluntary, means it's in our control. So whenever we want to stop the defecation, we can, whenever we can continue, we can. So it has a capacity to be in our control, only external, but internal is always in a parasympathetic and sympathetic stimulus. Now let us see the pathway, the distance of rectum. Actually here what happens is, now the pathway of this defecation reflexes, first from the rectum the reflexes come, the efferents come to the center, that is the sacral segment of spinal cord. So now it activates the sympathetic nervous system. So because of this there is relaxation of internal anal sphincter and also relaxation of external anal sphincter, but it's in our convenient time or environment. So whenever it's convenient, then we will relax the external anal sphincter, finally leading to defecation. So the rectal pressure, if the rectal pressure is 18 millimeters of mercury, then it is first urge to defecation. But if the rectal urge has increased to 15.5 millimeters, it is called soiling, means there is relaxation of both internal and external anal sphincter, means both sphincters are relaxed. If both sphincters are relaxed, means there is you can't control it, so it's a defecation, it's a not controllable defecation. Now let us see the migratory motor complex. It's also called MMC. So it is seen during fasting stage, so it's beneficial. So whenever a person is doing fasting, then all this bad material, undigested foreign material, anything, the waste thing which is present in the stomach will get out. So it's good to do fasting, it's beneficial. So all important contraction originates in stomach, migrates all the way and ends only in the colon. So the functions are, so remember, an important contraction originates in stomach, migrates all the way and enters only in the and ends only in the colon. So at the colon it stops, and from the colon again, by means of mass movements or by means of defecation reflex, finally it gets defecation. So till colon there is a strong movement, so that all the waste from our body will be reaching there. So functions, it's sweeper of the GIT, so it's like sweeping, cleaning our GIT, clean the GIT, especially foreign bodies, undigested food, bacteria, preparation of GI tract food, and also prepares the GI for next meal, means it is cleaning nicely. So actually one migratory motor complex is for every 90 minutes, that is 5 cm per minute, and it's regulated by motilin. So motilin is a timekeeper hormone. So motilin regulates this MMC movements. It is for once every 90 minutes, and you can see the three phases in this migratory motor complex. The first phase is no activity or quiescent period. The second phase is irregular activity. The third phase is burst of regular activity. So these are the three phases which are seen in the migratory motor complex. So this is like a very quick revision of this, because this if we'll do it slow and late, it will become longer video. So I did it very quick because it's a revision video, so you can understand faster.